Thank you for joining Track 3, Drug Development Pathway, sponsored by PRA Health Sciences. I am Cassandra, and I will be your host for the last breakout of Day 1. As a reminder, this track is being live streamed, and I would like to welcome our remote attendees and remind everyone to download the mobile app, which we will be using for the Q&A session. Our last breakout session will be presented by Mary Erickson, Senior Director of Medical Affairs and External Research at Intercept, and James Valentine, Associate at Hyman, Phelps, and McNarma. They will be leading our session incorporating real-world evidence into drug development and approval. Please join me in welcoming Mary and James.
Well, there we are. Good afternoon, everyone. As was just said, I am James Valentine. I'm an associate attorney at a boutique food and drug law firm in Washington, D.C. And I will be your first presenter this afternoon in our session on incorporating real-world evidence into drug development and approval. This is real-world evidence. If you're here, it's been quite the buzzword, probably just below CRISPR and patient-focused drug development. Real-world evidence probably has been the next most popular word being thrown around. So we're going to be here with you spending the next hour exploring what this term means and what roles it might play in supporting drug development and approval specifically. I'm going to start out providing you a bit of an overview and some background, and then Mary's going to take over and talk about some practical examples that Intercept has with use of real-world evidence. So in my presentation, I'm going to start out by giving you an overview of what is real-world evidence, how it has been used in research, what are some of its limitations, how it can be used in drug development, and importantly, as we're thinking about increasing use of real-world evidence, what are FDA's obligations regarding real-world evidence. So a lot of ground to cover. As we move through the presentation, we'll hopefully hit on some of the burning questions that you have, but we definitely want to save time at the end for any audience questions. So what is real-world evidence? There's many definitions out there. I'm a lawyer, so I turn to the law to define what it means. So in the 21st Century Cures Act, it actually defines real-world evidence as information on healthcare that is outside typical clinical research settings. So you're looking at information about one's health. It's coming from something other than clinical trials. So in other words, we're looking at information that is coming from routine care that's at least closely approximate to the eventual use in clinical practice. So we're looking at data that's coming not from a controlled trial, but instead the real world, which may seem obvious. But we'll talk a little bit about what exactly is routine clinical research, a clinical research setting. What might help in terms of understanding real-world evidence is to understand some of the examples of where real-world evidence has been generated from. So some examples are electronic health records, so information collected in the routine course of care that's entered in the physician or hospital setting most often. It also can include claims and billing data, so information that's collected to support reimbursement from insurers, both public insurers as well as private insurers. Information can also be collected through registries, both specific disease registries, like many of the patient organizations that are here are either running, or many are, I know, interested in running, as well as certain product registries for individual medical products. And lastly, kind of there's this broader and newer source of real-world evidence, which comes from information about health that is generated kind of through the normal course of a patient's day, whether that be on health-specific apps or just through social media. So these are all ways that outside of clinical research, data is being generated through the real world and what we can potentially use to help inform drug development. So these are often large data sets, and as all of these examples highlight, they were collected not for the purpose of assessing whether a drug works, but for some other purpose. So to help continue to find real-world evidence, we can contrast that with a traditional clinical trial, which is essentially what the 21st Century Cures Act defines it by exclusion. If you participated in the sessions earlier today in this track, you'll know that traditional clinical trials are conducted with specific populations. These are in very specialized environments that are very different from clinical care. They're very much more controlled. The control is there in order to ensure consistency and ensure quality in the data that's generated from trials. The way that trials do this is through, as many of you that may have participated or been involved in helping look at a protocol or even informed consent forms know, there's long lists of eligibility criteria. As part of clinical trials, there's detailed case report forms. This is a separate thing from an electronic health record, but is instead something that collects information that's specific to the trial. There's intensive monitoring during clinical studies that is not necessarily part of routine clinical care. Also, the staff that are conducting the clinical trials are often specialized research personnel and not the same practitioners that you would see in routine care. And lastly, there's a well-defined protocol that specifies what the dosing regimen will look like, if there needs to be adjustments in how the care is being provided, whereas this is all done on a physician and patient-specific basis during routine care. So lots of differences between a clinical trial and routine care that might generate real-world evidence. These traditional clinical trials, they're the primary way that we get information to support drug approvals. Because of all of this controlled variability, we feel very confident that what we're seeing is not confounded and not due to bias. However, one of the biggest critiques of a traditional clinical trial is that it really doesn't extrapolate to the real world. How can we generalize if we're restricting the population, restricting how care is going to be given to this very unrealistic care setting? How can we know that whatever benefits and safety risks that we're seeing in the course of a trial is really going to translate to the real world? So while traditionally real-world evidence has not been used to support drug approvals, it's not a completely new concept. It's something that's been used in healthcare research for quite some time. And so up to this point, related to drug development, we've used real-world evidence even. We use it for generating hypotheses about therapeutic development, so identifying what are the research questions that we want to ask when we do run a clinical trial. We can look to some of the databases of health information out there to see where there might be unmet needs. We use it for outcomes research. This is looking to see how healthcare systems or products in the real world are actually performing. We use it to assess patient care. So we might look at how different treatment protocols that are being implemented in different healthcare settings are performing. And that includes for assessing healthcare systems as well as for assessing particular interventions. Often in healthcare, we use it for quality improvement, finding ways to tweak the way that care is provided and see if there's ways that we can implement quality improvements to improve patient safety or other aspects of patient care. We also use it for safety surveillance. This is something that in the FDA world, real-world evidence has been used quite more frequently. FDA has the Sentinel Initiative, which is a program that is pulling together for the first time some of these different real-world evidence sources to see if there's ways to detect safety signals for products that are approved and on the market in ways that otherwise might not be caught and reported to the agency for their safety monitoring. And then ultimately, they can also help inform the design of some of our well-controlled trials. In general, also, even outside of drug development, real-world evidence is used can assess different types of clinicians, different kinds of healthcare settings. Just in general, any characteristic of a healthcare system, you can go and pull data and compare and do some observational research to see if there's differences. So that's traditionally how real-world evidence has been used. It's very efficient. It's often done retrospectively, so you're looking at data that's already there. You're not going out and collecting new data. And so this can save a lot of time and money from running controlled studies. So you might be wondering if there's all these benefits. It's quick, easy to do research using real-world evidence. Why don't we use it to support drug approvals? Well, there's a lot of flaws in the data itself with real-world evidence. All of the things that we've mentioned, from electronic health records to claims data, they're not collected and organized in order to support research. They're collected for other purposes. So they're not optimized for that purpose. They don't necessarily include the types of outcomes that you would look for to support a drug approval. They're not, the types of outcomes aren't those that might best measure, or most sensitively measure a treatment effect in a drug. And so it's not really fit for that purpose. We have some concerns about the accuracy and reliability of this data. Not every setting, for example, not every clinician's office has the same level of rigor in how it enters information into an electronic health record. That's just a simple example, but there's a lot of questions about the completeness of these records and the standards in which the records are implemented. And as I mentioned, this type of research is often done retrospectively rather than prospective like a clinical trial would be, although not all the time. But when it is retrospective, there are some issues that arise related to bias. And so the result is that, in general, real-world evidence is thought to be, have fundamental issues of confounding, data quality issues, and bias. And I think what might be more of an issue that we really need to think about as we're looking towards real-world evidence is, I think we kind of have this idea that when we're testing a drug, we're going to expect to see a dramatic effect, when in reality, drugs are approved off of somewhat marginal treatment effects in studies. And so if we are using real-world evidence, we need to make sure that we're optimizing it so that way we can see those differences between those patients that are being treated with an investigational product and those that might be on standard of care or placebo or the like. But still, there's, I think, good arguments out there for exploring the use of real-world evidence. The expense of traditional clinical trials just keeps going up and up every year. I think it's a safe argument to say that the clinical trials, despite being more expensive, aren't giving us necessarily more useful information than they had, in the past. So there's definitely a cost, especially with increased focus on the cost of medicines, looking for ways to minimize the development cost. Commissioner Gottlieb has definitely taken an approach where he's looking to see how the FDA can reduce the cost of development in order to translate into cost savings for patients. And unlike clinical trials, which is, by design is real-world evidence is more generalizable. And so you have an opportunity to actually understand, using real-world evidence, how your investigational treatment is going to work in the broader range of patients, those that might have been excluded from a clinical trial because they have some comorbidity or they're on some other drug, as well as you can use it to assess what adherence will be. If you're using assessing more investigational products in a more real-life environment, a real-life clinical setting, you can actually see if in real life physicians and their patients are going to be able to adhere to the treatment regimens as well as monitoring programs. And the electronic data is getting better. There's better systems being set up to support kind of universal standards as well as interoperability between these systems. And there's greater access to that better data. So there's real opportunity here. FDA has, while not having yet put out any guidance on real-world evidence, has started to give us some of its thinking on what it would like to see for real-world evidence to be a little bit more robust to support drug approvals. What FDA has asked for is ideally standardized data structure. linkage and that interoperability between the different healthcare settings and the different sources of this real-world evidence. One issue is they want to make sure that we have open access so that we're able to assess all of the patients that might be in these databases of real-world evidence. But to do that, we need to make sure that we have consent for full use in a de-identified fashion. And finally, really, we also want to make sure that some of these endpoints of interest, the types of outcomes that we expect to see a benefit of a medical product in, make sure those are incorporated into these the systems that are generating the real-world evidence, so that way we have relevant outcome information. So, despite some of the, FDA's wish list of what they would like to see, there definitely are opportunities in the short term to use real-world evidence. Kind of the first and most low-hanging fruit is that we can use real-world evidence to start answering questions of drugs that have already been approved. So once the drug has been shown to be safe and effective, at least in the clinical trial setting, we can start to then use these systems already in place to look at optimal dosing, to look at longer-term outcomes. Clinical trials also by design are short, and patients are going to be on drug much longer in the real world, so we can assess that. There are, when you go out into the real world, you're also going to be giving the drug to much larger patient populations, so you can start to have enough data to start parsing out different subpopulations and seeing how outcomes vary in subpopulations. And then again, you can look to see what side effects that might exist when a drug is either taken with other medications or taken by a patient who has some comorbidity and it wasn't studied, they were not included in clinical trials. That's kind of the boring use of real-world evidence. What's more exciting is thinking about how can we actually use real-world evidence prior to a drug's approval in order to support a demonstration that the drug is effective. And so, again, here's kind of what is thought currently to be the lowest hanging fruit, which is to run what are called interventional pragmatic studies. So this is a clinical trial of sorts, but it's leveraging all of the efficiencies of real-world evidence. So here, in this type of study that we're talking about, you have you're going to use a study environment that's closer to real life. So that includes looking at more of a representative population of patients that will ultimately receive the drug, or you intend to have received the drug. The intervention is going to be used in a way that might be more similar to how it would be used in practice. Your comparator might not be a placebo, it might be much more of a standard of care. And it's not going to be necessarily as rigorous of a standard of care, not as set. You're going to allow physicians to modify standard of care as they would in clinical practice. And then, again, you're going to try to leverage outcomes that already exist in the real world. But one of the benefits of doing this, even though kind of all of that sounds like all of the things that we said might undermine the strength of a study, is that we're going to have a protocol that pre-specifies and exactly who is going to be involved in the trial and what it is we're going to be looking at. And so, unlike doing a retrospective look at existing data, here you're able to minimize some of that bias that FDA is concerned about. Also, by prospectively enrolling, you're able to ensure that you're not having any kind of recruitment bias in terms of the patients that are participating. We know who's going to be participating. We're not cherry picking. And so one way that such a trial could be made more robust, it might not be feasible to do patient-level randomization. So each patient that comes into a clinic setting, the physician then has to, have that patient be randomized to either the intervention or the standard of care. Instead, you might be able to randomize entire clinics or hospitals or other care settings. So if you have, if you identify 20 physician groups that you want to include in this pragmatic study, you randomize 10 to implement the intervention and 10 to implement standard of care. And then you do your comparisons between those groups rather than randomization on a patient level. And so one way that FDA has kind of hinted to that this type of pragmatic study might be used is not to replace the randomized controlled clinical trial, but really to supplement it. So if you run it in parallel to a randomized clinical trial, you can then look and see if there's trends or similarities in the results that match up, in which case this real world data can help corroborate what you're seeing, and you can feel confident in terms of the generalizability of what you found in the kind of more tightly controlled clinical trial. Something that hasn't really been talked much about, but I think is really relevant to this audience in the rare disease community, both drug developers and patient organizations have really taken it on to help kind of the field understand the natural history of their diseases. And so I think it would be a missed opportunity not to look to see how these natural history studies, and even in some cases more simple registries, can be used to provide real-world evidence that can then be incorporated into a drug development program. So the way that I would propose this be used is you generate natural history data in the course of a natural history study, and you use that as a historical or an external control to a clinical trial. So you run your clinical trial like you would. You have your interventional arm, and perhaps you even have a small control arm, a placebo arm or a standard of care arm that's randomized. And then what you do is you, at the same time, use your natural history data to be a control outside of the study. So you're applying the same standard of care to as those that are in the control arm within the study. And then what happens is at the end of the study period, you consider kind of doing a mixed model control, where you have both those internal to the study that were the randomized at the patient level, perhaps because you know you're going to be supplementing the control arm with this real-world evidence control. You can do a smaller control arm within the study. Maybe you randomize 2-to-1 on drug and control, or 3-to-1. And then you kind of pool all of those patients, both in the placebo or standard of care arm in the study and those in the natural history study, to kind of increase the power of your study to be able to see those between group differences on the outcomes that you're looking for. It really might help increase, and especially in rare diseases where we don't have always the ability to run larger studies, create more power, but kind of avoid some of FDA's traditional concerns with external controls, because you're able to then look both at the data from inside the study as well as that outside to see if your control arms at least have a similar kind of trajectory in the outcomes that you're looking at, to feel confident that you are really looking at similar populations of patients. And FDA has historically exercised flexibility in allowing these types of historical controls. So here, perhaps this kind of mixed model is something new that we should think about, at least, in addition to kind of the pragmatic trial design. So I'll quickly mention that FDA, while this is something that is of interest to many stakeholders, because of that, FDA now has some requirements under recent laws to explore real-world evidence. Under 21st Century Cures Act, which was enacted in December of last year, FDA has to look at how real-world evidence can be used to support new indications of approved drugs, as well as to satisfy post-approval requirements related to safety. FDA has to develop a framework for this in 3 years and issue guidance within 5 years on this framework. Importantly, this law says that the standards for approval have not been changed. And then under the more recent reauthorization of the Prescription Drug User Fee Act, FDA has committed to enhancing its use of real-world evidence in regulatory decision-making. It's not as narrow in focus, so it could include the approval of new drugs. And so in order to advance this goal, FDA is committed to hosting at least one public meeting by the end of next fiscal year, as well as to form pilot studies and other methodology kind of development projects to address some of the concerns that are raised at the public workshop. And so there's really the PDUFA commitments and the 21st Century Cure Act requirements are likely going to be kind of one cohesive plan that FDA is going to move forward with to explore real-world evidence. FDA has already started doing this. They are working with the National Academies of Science, Engineering, and Medicine's Forum on Drug Development, or Discovery, Development, and Translation. Sorry for all of those words, but they're hosting 3 workshops over the next 2 years. One is already scheduled for later this month, really looking more at incentives. How can we incentivize the entire ecosystem to invest in real-world evidence? But then a couple of workshops next year that really get into the nuts and bolts of how can real-world evidence be used and what are the methods for doing so. I'll leave you with an example of a pragmatic study in real-world evidence. This isn't all theoretical. It's actually being done. In May, GSK put out a press release that reported the outcomes of a pragmatic study that they were doing of a asthma drug of theirs. This was an open-label randomized study that was done in the UK to confirm the benefits of 2 different doses of one of their inhalers. This was done in a very pragmatic way. There was minimal exclusion criteria, and the physicians were allowed to treat patients as they would normally. That was not set in the protocol. The only restriction was you couldn't switch someone from standard of care over to the intervention. They looked at disease symptoms that were key to controlling asthma, the wheezing and tightness. So they did follow kind of FDA's ideal of including outcomes that would be of interest to assessing benefit. Ultimately, although this is real-world evidence, it wasn't an easy undertaking. This involved over 4,000 patients. As you can see, many sites and many pharmacists had to be involved for this to successfully be run. But ultimately, they were successful at showing the benefit of their product in the real-world setting, confirming the benefits that they had seen in clinical studies. I just leave this since I know you can download the slides. FDA hasn't put out guidance yet, but they have published a couple of articles. Here's the sites for those. If you're interested in the FDA's commitments and requirements under cures and PDUFA, here's some links to download some summaries that we put together. So with that, I thank you, and I'll turn it over to Mary to talk about some real examples.
Thank you, James. I appreciate it. Oh, did I just, oh, sorry. I think I just reset. Oh, thank you. Okay, so as James said, I am here to give you some real-life examples from the experience here at Intercept Pharmaceuticals. So as I enter my talk, I'm going to just briefly tell you who is Intercept Pharmaceuticals. Then I'll give you an example of a surrogate endpoint in a disease called primary biliary cholangitis, or PBC. And then I will wrap up my talk with an example of a patient registry in another disease that we're studying, and that is Primarysclerosing cholangitis, or PSC. I just need to go ahead and show the disclaimer slide that says this presentation is for educational purposes only and not to be reproduced. So who is Intercept Pharmaceuticals? We are a company that was founded in 2002 by our CEO, Mark Przansky, based on his belief in the promise of bile acid therapeutics for non-viral progressive liver disease. As I mentioned, there are several orphan liver diseases that are in our portfolio that we're studying. They are indeed PBC, as I mentioned, PSC, and then biliary atresia, which I will not talk about today. So let's go ahead, in the interest of time, jump right into our first example, which is establishing a surrogate endpoint in primary biliary cholangitis. So what is a surrogate endpoint? A surrogate endpoint is a substitute for hard clinical outcomes. Since we're in liver disease, those hard clinical outcomes are going to be things such as liver transplant and death. So I think you also need to understand that those surrogate markers are typically blood-based. They're typically things that you can measure, such as liver enzymes, and those have been really critical in this program. So I'd also like to just give you a little bit more information about what PBC is, so that you can understand how this really impacted us. PBC is a rare autoimmune cholestatic liver disease. It's commonly found in women at about a 10-to-1 ratio over men. It's characterized by the destruction of intrahepatic bile ducts, and it leads to cirrhosis. So as I said, it leads to that hardening of the liver that then causes, obviously, functional issues with the liver, and then you need to have a liver transplant. Its symptoms prominently are fatigue and pruritus. And it can be associated with other features as well, such as increases in cholesterol and osteoporosis. Today, there are approximately 47,000 people in the US who have PBC. Those are people that's actually in the ethnicity of white. We do actually at Intercept have ongoing efforts to understand additional epidemiology in the US as it relates to other ethnicities, and we hope that data is published very soon. Again, this slide just goes through in more detail the pathogenesis of the disease. I think important aspects from this slide for you to take away are the facts that this disease is not only environmentally caused, it also has a genetic basis relevant to global genes. And also the fact that this is a multi-stage disease. It has a silent phase, and then it goes into phases where you see rises in ALP and bilirubin. Those are important markers as we talk about the data that we used to support our development program. And the other aspect here is that there is a variety in how the disease progresses in patients. So some patients, it's really a slow progressive disease, and other patients, it can progress faster. There was a breakthrough in PBC in 1997, and that was the approval of the bile acid ursodeoxycholic acid, or UDCA. This is fabulous, as it really indeed helped many patients. However, as you'll see from the guidelines that are shown on this slide, there wasn't actually a specific definition of treatment success. And there were a lot of publications done from a lot of different groups and a lot of investigators on small populations of PBC patients, and they had different measures of treatment success. But even as you looked across all those measures of treatment success, there were still about a 40% of patients didn't really seem to respond adequately to UDCA, and some are even intolerant to the molecule. Thus, there was a large unmet need, and Intercept stepped in to develop obeticholic acid in this space. However, one of the big challenges, as I said, was that you needed a surrogate endpoint to really speed the development of this new medicine and be able to get this to patients in a faster way. That's because this was, again, a slowly progressive disease with low prevalence. So the two markers that were out there that were candidates were, as I mentioned, alkaline phosphatase, ALP, and bilirubin. But the question was, where were we gonna get a large data set to be able to then retrospectively look at those patients' records and determine if those were indeed surrogate markers that could be used in our approval and in our trials. So David Shapiro, our chief medical officer in 2010, really went out and said, I want to work with the experts in the world. As I said, there were multiple centers. They're on this map. They agreed to meet with David and collaborate together. This was a big win. In 2011, Intercept provided a grant to support basically a meta-analysis that would be performed by what was then known as the Global PBC Study Group. This slide actually shows you the people that were involved in this Global PBC Study Group. It was really run out of Erasmus Medical Center in Rotterdam, Netherlands. In the middle of the picture, the woman who's blonde is Bettina Hansen. She was the lead statistician on this study, and then all the really leading PBC investigators collaborated and provided those records so that, as James was talking about, really what happened here is you could go back, you could look at those charts, you could make sure you had all the data on those patients over a long period of time, put that in a central database so that it was indeed structured and could be statistically analyzed. And in 2014, the sort of capstone of this was that in gastroenterology, Lammers published a paper that did in fact show higher ALP levels resulted in reduced transplant-free survival. So you knew that at baseline, if you had higher ALP levels, that your chances of progressing to a liver transplant were higher. This was obviously a pivotal moment for us and allowed the design of a phase 3 clinical study that used ALP and bilirubin as surrogate endpoints. It was under that program that we were able to indeed receive accelerated approval for obeticholic acid in 2016. So this is, just to keep in mind, this is an accelerated approval, so we do still have ongoing an outcomes trial that will actually complete the program and needs to be done. But what this allowed was the access now of patients to a new medicine, the first medicine in really 20 years since 1997. So again, real-world data was critical to the success of bringing this new medicine to patients. So what are we doing with real-world evidence now that we actually have a new product for the treatment of PBC? We have actually partnered with a company called Target Pharma Solutions. Target Pharma Solutions has set up a real-world observational cohort. They launched it in October 2016. They're collecting both retrospective and prospective data. They plan on enrolling approximately 1,500 patients across 50 sites in the US. They have today approximately 250 patients. This is critical for us as this is a way to understand how patients are doing, and they will send out surveys. They can do patient-reported outcomes because these patients are consented. It also provides continued safety data for patients who didn't meet the criteria for our strict clinical trials. Target Pharma Solutions actually also has other cohorts in other liver diseases if you wanted to look them up. So the other disease I want to talk about today and is sort of looking to the future is primary sclerosing cholangitis. This is again an orphan liver disease that is rare, and it is often called the brother disease to PBC. Again, you have a progressive disorder of unknown etiology that leads to the destruction of bile ducts. And in this case, there's actually currently no approved therapeutic for patients with PSC. This, unlike PBC that affects mostly women, I said it affects mostly men. 65% of PSC patients are men. And again, we have a situation where it's a slow progressive disease, and so a surrogate marker is again relevant, especially because the epidemiology shows you that there are fewer cases of this than PBC. Intercept did just release several months ago top-line data from a phase 2 study called ASAP, where we looked at obeticholic acid in PSC. We were able to show that obeticholic acid did lower ALP levels, so alkaline phosphatase again as a marker in this disease at 24 weeks, and we did have pruritus as the most common adverse event. But this is exciting, and this means that what we need to do is look at, if we're moving to phase 3, how we will do that. This is a great time to actually engage patient advocacy groups. And the next part of my presentation, I would like to thank Rachel Gommel and Ricky Safer from PSC Partners for providing these slides. They're instrumental in having a patient registry and being able to work with us and the FDA in terms of how these clinical trials move forward. So just a little bit about this registry. It is different than the examples I talked about previously because this is actually a patient-generated and a patient-driven database. So PSC Partners was founded in 2005, but they got into this registry in 2010 at a NIH workshop for rare diseases. They then have been working with the FDA and with pharmaceutical companies ever since. This is HIPAA compliant and FSMA compliant as well. And it is monitored annually by an IRB. I think the other key aspect of this is that just like the PBC example where we worked with the experts in the field, PSC Partners is working with Chris Bolus to ensure that this database is designed in such a way that it's patient-friendly, but yet then it actually also provides data that are helpful to see whether patients might meet inclusion-exclusion criteria if they wanted to be involved in any clinical trials. So this is just a little bit more about the operations of the registry. Patients enter their information and they have to enter proof of diagnosis. They also are consenting, so that's helpful in terms of being able to reach out to those patients and have them complete surveys. But importantly, the identifying data never leaves the registry. Today, there are 1,076 participants in the registry. Most of them are from the US, but this is a worldwide database. And again, this is an example where patients from all over the world are able to contribute, and so it gives you a more real-world snapshot of patients and their disease state. It does also offer, as I said, a pool of patients who might be interested in participating in clinical trials. So again, this speeds the research process. It also builds awareness of the disease and allows collaboration with both the FDA and pharmaceutical companies. So as a summary to my presentation, I'd just like to say that we provided you an example of how real-world data from our global PBC supergroup was critical in bringing forth a new medicine for PBC patients, the first in 20 years. We continue to invest in registries that will answer critical questions for PBC patients. We have strong partnerships with patient advocacy organizations, including the PBCers organization and the PSC Partners. And again, I just wanted to thank Rachel and Ricky for their slides. Basically, we will continue to invest in real-world data in the future for our development programs. I also wanted to just wrap up with sort of the fact that these are 3 different examples of how you can use real-world data. You can combine existing data sources. You can look to partner with a large company that's already doing that, or you can look to start your own patient registry. And with that, I will hand it back over to our moderator.
Cassandra apologizes. She had to step out for a minute, so I have her moderator app. I have her phone, so if anyone online would like to ask questions, I can take those from here and then also from the audience. I see that someone has their hand up there.
Yeah, so if you look at the pipeline page of our website, we are looking at obeticholic acid as a treatment for NASH. They have none either, yes. That's not an orphan disease, but yes, there is no treatment for NASH, yes.
Were there any other questions? Oh, yes.
Yeah, so the question was related to one kind of potential option for using real-world evidence, particularly the pragmatic studies, and doing those in parallel with randomized controlled trials, and how that might actually be counterintuitive to the idea of spending less money on drug development. So I think kind of looking forward to the future, one of the benefits of doing the pragmatic study might be immediate. If you do a single study, that's a traditional clinical trial, and then you have this pragmatic trial, you might be able to have that form the basis of an approval, saving you from needing to do a second traditional clinical study. Obviously, there's no precedent for that yet, but there are precedents for other kind of single study approvals where you only have one traditional clinical trial, but then you have something else. There's this regulatory term is confirmatory evidence. It's not really defined, but I think it's certainly broad enough to include data generated, especially from a prospective pragmatic study. So I think in some ways that would save money because that second study, the pragmatic study, would be less expensive than a second traditional clinical trial. Perhaps as there's more experience with those types and other types of real-world evidence-based prospective studies, there'll be more comfort at FDA, and they might find other ways to kind of expedite approval using those. But I don't think we're at a place where we're going to totally replace the traditional clinical study. I think you're always going to need at least one of those, but what might happen is you might be able to incorporate some elements of a pragmatic study or real-world evidence into that to maybe reduce the burden of administering that single study. But I think that's kind of definitely more in the future. I think now the idea of using it as complementary and still supporting your regulatory approval would be kind of the goal.
Yes.
So yeah, it is used in conjunction with UDCA, or in patients who are intolerant to UDCA, it can be used alone.
Yeah. So the question was, and I don't know what organization you're from, but you recently launched a patient registry for PKU, and they're having some trouble getting historical information from patients uploaded. And so the question was, would that information still be useful to FDA or to drug development? And so I think the answer is yes. It all depends on what the use of that data is going to be. I think early in my slides kind of included this whole list of different ways that real-world evidence can be used in drug development. I think having that even incomplete historical information provided by patients is really valuable, because in rare diseases, there's not much in the medical literature about the disease. So you need to rely on what you have, and absolutely drug developers and FDA would be interested in that. But in terms of that type of, one, historical, but two, also kind of heavily maybe incomplete data, I don't know that that would be high enough quality to serve as a control for a clinical trial, kind of in that whole slide where I talked about using natural history studies to supplement control arms.
Are there any other questions?
I haven't gotten any that have come through the app. Okay. With that, thank you, Mary and James. What a great takeaways and insights into this topic. This concludes this afternoon's breakout session, and at this time, we ask for you to jump on the mobile app and take our brief survey. We would also like to invite you to stay for the documentary screening and discussion on the story of hope and persistence by We Carry Kevin. Their presentation will be followed by a backyard party and reception sponsored by PRA Health Sciences, where you can participate in a collaborative puzzle art project. Thanks again, and let's give Mary and James a last round of applause. Sorry.